So welcome everyone. My name is Radek and I'm a PhD student in the group of Radoslaw. And today I will talk about my project, which is about single molecule microscopy in zebrafish embryos. So not to take any time longer for the introduction, I'm just gonna move to the topic. So first of all, I would like to start from presenting the standard single molecule microscopy setup. So basically we have a sample which has fluorophores most often fused genetically to the protein of molecule of interest, and those fluorophores are excited by the laser beam. And then thanks to the phenomenon of the Stokes shifts, when they go to the relaxation state, they emit the photons at the longer wavelength, which can pass through the dichroic mirror and is detected by the camera device. And generally, the single molecule microscopy setups consists of objectives with high numerical aperture with increased photon collection capability. So now, when we know how the single molecule microscopy setup looks like, we can move forward to what we can actually do when studying molecular dynamics with single molecule microscopy setups. So as you probably know, with single molecule microscopy, we don't take static pictures, but we observe the order of events when it comes to dynamics of certain proteins or molecules with a high spatial and temporal resolution. So spatial resolution is high enough to allow us to observe the single molecules over time, and temporal resolution in this case, for example, you can see is 10 milliseconds. So we can actually take pictures at a very fast time point. So we can actually see what happens with the molecules at really small intervals of time. And thanks to that, we can observe what happens with those molecules and how do they behave over time. And with this, I can apply those techniques and those abilities of single molecule microscopy to study the mobility patterns of molecules in living zebrafish embryos. So now I would like to talk about the experimental setup. So how do we actually do it in the living zebrafish embryos? So this is the standard experimental design for imaging living zebrafish embryos using SMM techniques. So basically, at the 0 to 1 cell stage zebrafish embryo, we inject our embryo with the molecule of interest, which can be DNA or mRNA microinjections. And this molecule is genetically fused, as I mentioned before, with the fluorophore of interest, GFP, YFP, or others. One day post injection, we screen the molecules to look for to check if we have the expression patterns that we would like to see. And then with the positive screening of our molecules, we can move forward day after to actually to proceed to the single molecule microscopy imaging. So so when the fish is also more developed and easier to mount and image. So now we have the single molecule microscopy setup, and we have the model, and we have the experimental setup. So what actually we image using our setup? So basically, our protein of interest is HRAS. HRAS is the protein of the RAS family, and it's a small GTPase that is a key early player in the signaling pathway leading to cell proliferation and division. And it's shares, there are a lot of isoforms of RAS proteins that differ mostly in the hyper variable region at the C-terminus end of the protein, and the most carboxyl terminal part consists of the anchor domain that is responsible for anchoring the protein at the inner side of the phospholipid or the plasma membrane by post-translational modification and addition of lipids to the protein. At the N-terminus, as you can see, we have genetically fused the YFP protein, which we will be able to see under the single molecule microscopy setup. HRAS it was reported before that 25% of the genetic mutations in HRAS are reported in 25% of cancers present in humans. So this makes it protein a very interesting target of cancer therapies. So maybe the aspect of differing dynamics in mutants may play an important role in understanding the mechanism of cancer origination and spread. So this is our molecule now. So that's why we also selected this molecule. And the research questions considering what we want to see in HRAS are as follows. So first two questions we want to see is, do we have the one fraction of the molecule in the membrane, or do we actually have more fractions in the membrane? And what is the percentage of those fractions? So how many molecules belong to one fraction, how many of them belong to the other fraction? And what we also want to see is if those molecules actually move within the lateral, within the entirety of the lateral membrane, or actually are confining to certain areas. So basically they are restricted, the movement is anyway restricted by certain obstacles. And that we do with one single molecule microscopy techniques, which I'm going to move to in the next slide. But then with the help of the other single molecule microscopy technique, we can also see if those molecules always belong to one fraction, or if they actually switch between fractions. So if this actually dynamics of protein is more complex than we thought before. So first, let's move to the first two questions that I mentioned, and that we do with the single molecule microscopy technique called TIRF, which is total internal reflection fluorescent microscopy. And here you can see the mounting setup of our TIRF microscopy technique. Well, the excitation laser beam is totally reflected and the interface of our cover glass sample, and that leads to the creation of the evanescent field. So basically the laser beam doesn't penetrate the sample like in the epifluorescent mode, but rather excites the very thin layer through the evanescent field creation. So only the proteins in the outer layer of our zebrafish epidermis are excited. And so basically we reduce any fluorescent signal coming from the deeper layers of the fish. And that's very important when you actually study the dynamics of the single molecules in the membranes of the zebrafish embryo. And this is also our mounting setup. So basically to push the tail towards the cover slip, we use the agarose sheet. So basically we limit any movement, and also the fish is anesthetized. So that guarantees the immobility of our embryo throughout the measurement. So this is the TIRF setup. And here I want to show you basically when you look back at the experimental design, I told you that at the first day we do the screening, pre-screening. So in this case, you see the confocal of the YFP HRAS molecule. So we clearly see that most of the signal is localized in the membranes of the fish. So then with this fish, we know that the injection worked, and then we move forward the next day to the SMM single molecule microscopy measurements, and then you see basically the typical picture that we get from the imaging membrane of the zebrafish embryo using the TIRF setup. So now we have those pictures. We collected the data, and what then happens? How do we actually analyze those fraction sizes, and how do we actually analyze the confinement areas? So that's what I want to explain right now. So with data analysis, first of all, the question is, do we have one fraction or two fractions? Are those molecules moving in the same way, uniform way, all of them, or they're actually differing in the diffusion coefficients or any other aspects? So that we describe by fraction size. So we actually don't track the molecules with our software. We actually plot the cumulative probability measurements based on the radius. So basically the cumulative probability tells us what is the probability of finding the molecule after 25 milliseconds in this case, because that relates to time lag. So we, after a certain time, what is the probability of finding the molecule at a certain distance? So you see that at a certain distance in this graph below, the probability is 100. So at some point we are sure that we will find the molecule in the next frame of our measurement, considering that the time lag might be 25 milliseconds. But, and then, sorry, when we plot this cumulative probability, so basically the probability of finding the molecule in the next frame, we can fit the mathematical model of one population, two population, or more population model. So as you see in this graph, one population model doesn't really fit well in our data, which is in red, but instead we fit it the two population model, which actually with which you see that in the equation, I just want to mention that in the equation you have elements as alpha and mean square displacement of fast and slow fraction. So now we fitted our data to the two population model we fit, and this population model gives us the information about the fraction size, square displacement of the fast fraction, and square displacement of the slow fraction. So we know what was the square displacement. So we know that our HRAS exists in two different fractions with differing square displacements. right? So we have now information about 3 parameters: fraction size, square displacement, and of each of the fractions. And what we do next? How do we then establish the confinement area or the initial diffusion coefficient? So basically, we plot those square displacement over time lag. So we see what happens with the square displacement with the increasing time lag. So there are 2 possibilities, right? So if the equation, if you see the graph below, if the trend is linear, if it's a linear correlation, then there's no confinement. So the molecules actually move freely and they are unconstrained by any factors, whether it be genetic or structural factors of the cell. Otherwise, if you see the saturation of the graph, you see that at certain time, molecules are not moving any further, and that means that there's something that blocks them from moving any further, so they're actually confined into microdomains, right? So with that, we can establish, and when we feed the confinement model to those saturated plots, we can then extract the information about the confinement size and the diffusion coefficient of each of the fractions. So in the end, we report our results by reporting the fraction size, diffusion coefficient of every fraction, and confinement area of every fraction. And then with that data, we can use different genetic mutations of the protein. We can study the molecules dynamics at different developmental stages of the zebrafish, and we can then see what happens when we image different molecules or genetic variants on environmental stages or under stress conditions, and we can then see if there are any factors that influence our dynamics and confinement areas and diffusion coefficients. So here, I would like to show you one graph that we established for the HRAS wild type, and it's constitutively active and inactive mutant. Constitutively active mutant is the point mutation at position 12. It's called V12, and inactive is the point mutation at F17 position, and it's called abbreviated as N17. So here you see we have 2 fractions and they are differing in the diffusion coefficient. But what is interesting, if you look at the right graph, you see that the slow diffusing fraction, because this corresponds to the slow diffusing fractions, they're all confining. And that was reported that they are in the lipid rafts, or actually the fast diffusing fraction were in the non-cholesterol enriched domain. So they actually aggregate because they're in the membrane, as I mentioned. They play a role in the signaling pathway. So they need to enhance the response that comes from the extracellular membrane to pass the signal further in the signaling pathway. But what was not observed before is presented on the graph on the left side, meaning that before, in the studies performed on cells and HRAS, it was reported that those molecules are moving unconstrained. So what we showed using our analytical tools and microscopy techniques, that those molecules actually also confine to certain areas. And what is also interesting, that the confinement area of the constitutively active mutants is significantly higher than for the constitutively inactive and wild type HRAS. And that hasn't been reported before. So we basically show that those molecules in the fast fraction also confine, but they are not confining to any lipid rafts or very small structures, because the confinement areas are way bigger, which is also presented in the table, approximately 4 or 5 times bigger than the confinement areas for slow fraction. And that makes us think that there are other components of the membrane that constrain those movement of the molecules. So those types of discoveries that we make haven't been reported before, and that opens a new field of research that can be obtained through single molecule microscopy techniques. So now we know there are other constraints that also limit the movement of our fast fraction, and that requires also studies of the single particle. So how do we do these studies on the single particle? We move to another technique called two-photon excitation fluorescence microscopy, which is based on excitation of the fluorophore by 2 photons simultaneously, let's say. They have to be excited in a shorter time than 1 femtosecond. So basically we can assume they are excited simultaneously by 2 photons. And you know, like in 1 photon technique, you have the excitation cone of light, and in 2 photon, you only excite the molecules at the focal plane, because the probability of two photons exciting the same molecule at the same time is very low. So basically, only in the middle of the cone of light, when you have the density of your photons very high, you excite your protein of interest, and that leads to absolutely no excitation in the out of focus planes, and that, as you see in the movie, we see less blinking, and that makes us also follow the single molecules over a longer period of time than in TERF. So we just started this. We just recently started this project to look into the single molecules thanks to the two-photon excitation fluorescence microscopy technique. And now, with the help of students and other people at Leiden University, we locate those molecules, and we locate the single tracks, and we look, for example, this is just what we started doing. We select the tracks over single molecules that we can observe over a longer period of time, and we see what happens with the diffusion. So here, as you see, for example, on the right side, you see that those molecules, for example, they have this abnormal type of movement, and they jumping at some point, or they might even switch the diffusion coefficient. So now what we tend to believe is that those molecules are not actually always in the same fraction, but they can also switch depending on their circumstances, or we don't know yet what, but they actually can switch between the fractions, and they are not always constrained by the fraction. So I mean, also sounds plausible that there's not the same molecule cannot be always in certain fraction, and what determines which molecule is in which fraction, they actually switch between the states. So I would tend to describe them as the molecular dynamic states of the same molecule rather than fractions. So that's what we are doing now. But just not to prolong any further, I want to conclude what we did in my research and this presentation. So what we already shown, that the HRAS molecule has two fractions, because that was done in the TERF setup, a fast and slow diffusing one, and they all confined. And what was interesting also, that the fast fraction of the molecule is confined to in the microdomain, not microdomains within the membrane, and that are not based on lipid rafts or any small confined microdomains that are responsible for enhancing the signal coming from the extracellular matrix. And we also did the study characterizing the model, so the Zebrafish study. So we showed, I didn't show the data because there's no time, but we showed that there's no difference when we image the dynamic states at the different developmental stages of the fish from, let's say, from 1 to 2 day old or 3 days post-fertilization stage. So that's very good because the system is reliable. And then we saw that genetic mutations has some impact, a significant impact on the protein mobility dynamics. And what is interesting, that even in fast fraction, the constitutively active HRAS characterizes by the larger confinement areas. And what influences that movement, whether there are some structural components, whether this is the rearrangement of the cytoskeleton, that might be, that it's yet to be determined. But for now, with this, with the development of single molecule microscopy, we can now, with the two-photon as well, we can maybe start to depolymerize the parts of the cytoskeleton part of the cells to see if there's any impact, and observe them over the longer period of time to determine if the molecules actually expose different types of movement, if they are jumping, if there are certain abnormal behaviors, or if they are actually switching the states, and what is the time of what is the time between the switches of the states. So that's yet to be determined, but that was a synopsis of my PhD project. And thank you very much for your attention, and thanks to my supervisors, collaborators, students, and also collaborators at ICFO, where I did my secondment, and then we worked on the light sheet fluorescent microscopy, but that's the topic of another presentation. So thank you very much for your attention, and if you have any questions, please do not hesitate to ask me. Thanks.
Thank you, Radek. So any question? Radek, then I have one. Okay, you show, or you hypothesize that in fact, the proteins do move within microdomains.
Yes.
But what is surprising is that, okay, let's keep that. No, the previous slide was very interesting. Yes. Okay, but what is strange is that depending on the mutations that harbors the molecule, the microdomain doesn't have the same size. Okay, so how is it possible that the size of the domain would be influenced by the exact nature of the protein sequence of your RAS protein?
Yeah, exactly. That is an interesting question, and I can tell you that when it comes to how it was explained before in the slow diffusing fractions, which were already reported to be confined, was that most of them actually confined to the lipid rafts, but the active mutant, which is loaded with GTP, I believe, or active HRAS, which is loaded with GTP, has a lower affinity to the lipid rafts, and instead is confined by the lipid independent microdomains. But what exactly was that, and why exactly also the fast fraction is confined? And I mean, I understand your question, that I would suppose that there's some role of the cytoskeleton rearrangements, but why actually the V12 has significantly different sizes of the confinement is an interesting question. But in the fast fraction, this is I don't know yet exactly.
Okay, so your hypothesis, in fact, is that the activity of the enzyme would change because of the activity of the enzyme. The physical barriers that do border this microdomain would change.
Yeah.
Okay. Would it be possible then to have a double system with two different diffusing molecules, for example, with two different colors, to see whether this mutation would change the diffusion microdomain of another protein?
Yeah, I mean, that is of course possible, right? And we also actually wanted I also aimed at some point to and that would be very interesting to see the for example, double stain of molecule and actin. But yeah, for example, but so far yeah, we haven't done it yet, but that's definitely something that would really enrich our knowledge on that. And I agree with that. That's in principle, it should be possible to see that, right? Maybe not at a certain time lag, because we would have to switch, you know, when we have to image two colors, then you have to switch the excitation beam, and that basically makes your measurement a bit prolonged, so not 25 milliseconds. So there's always some trade-off, right? Okay. But that would be definitely possible, for example, with Twophoton, to see what happens with the two colors at the same. Well, it will be switching between 1 and 2, so it won't be at the same time. But maybe there's something that could be done there. Yes, I agree with that.
Okay, thank you.